Welcome to this review on genetic engineering and biotechnology. This discussion will be framed within a concept that I'm sure we are all familiar with from our biology coursework, the central dogma. Information, as it flows from DNA to RNA to protein, can be interpreted in order to reveal specific insights about the system being measured. There are numerous important trends that underlie the flow of information that I wish to go over first. DNA in this model is represented by a book because it's analogous to a library of data that is read from. RNA is a version of Hermes, the traditional Greek and Roman messenger god who is capable of agency in a limited sense, but mostly transfers notes between different individuals. Proteins are represented by a hammer because they act directly on systems in order to influence events. The trend discussed in this slide involves variability. As the information flows down the chain, the diversity of final products increases as well. One DNA strand can become 5 different mRNAs that eventually become 10 different protein products. In this way, the variability in structure and function increases as you move down the chain. At the same time, specificity increases. The shape of a biochemical object determines in large part its function. Proteins have the most specific shapes because each protein is suited for one primary function. Permanence is the quality of an object to remain constant. DNA is the most permanent, as the lexicon of information is in an extremely stable chemical configuration and is transcribed over and over again, whether for cell duplication or to produce RNAs. While a select few RNAs have more stable configurations, most last little over a few hours before disintegrating. Proteins have a large range of stabilities, but they often need to be replaced regularly. Our capacity from a technological standpoint to measure the information decreases as we move through each of these stages. DNA sequencing is constantly improving and becoming less expensive. These are low latency, i.e. sequencing occurs quickly, and high throughput, which means that they very accurately record base pairs, sequences. The process has been adapted for RNA but is more costly and relies on the accurate transcription of RNA into cDNA. Protein sequencing has been conceptualized, but it is still reliant on antibody tests for specific proteins that are often high latency, i.e. sequencing occurs slowly and low throughput, limited accuracy. These are the 4 trends I have chosen to highlight because they form the technical basis for what is possible to do with genetic engineering today. I will now turn to some of the most important and widely utilized technologies within genetic engineering. DNA technology from the late 1980s and 1990s focused on modifying the genetic composition of organisms by adding extra bits of circular DNA called plasmids. These would be transcribed and thereby include specific proteins within the cytoplasm of the modified cell. While this process was easy to perform on simple organisms, scientists ran into difficulty when applying the technique to more complex creatures. Although I've already discussed briefly our capacity to measure information within cells, I've included this slide to underscore how our increasing technological capability for DNA sequencing is enhancing our understanding of the so-called library of life. Many scientists are spending their time trying to understand how different DNA sequences apply to the phenotypes, i.e. represented traits we see in organisms. CRISPR-Cas9 is the newest mechanism available to edit DNA sequences. It is a far easier process when compared to its predecessors. It allows nearly any sequence of DNA to be edited with a high degree of accuracy and has thus ushered in a new era of possibilities for genetic editing. RNAi, with the I standing for interference, is a kind of RNA technology primarily utilized to decrease levels of a certain protein. It is utilized in agriculture to create GMOs that are either resistant to certain pesticides or with different nutritional compositions that are believed to be healthier for humans. This technology has allowed scientists to understand better the complex signaling pathways regulating the many variable functions within organisms. We will now switch from discussing technologies to examining certain impacts of genetic engineering. Although I cannot discuss all of the examples on this slide, I would recommend that you on your own time read through this slide to understand the drastic impact of GMOs. In addition to monopolizing the consumption market and becoming the primary product in our grocery stores, GMOs also spread into the wild and outcompete native varieties. This decreases genetic diversity within the environment and leaves the species more vulnerable to novel infection. GMO salmon is the first approved GMO animal that can be sold within the United States. It was developed 30 years ago with a plasmid that allowed the salmon to grow much faster than native varieties. Although it has undergone 30 years of testing to ensure efficacy and cannot spread into wild populations, it is unclear whether future GMO animals will undergo the same rigorous testing. Furthermore, vendors are not required to include labels on their packaging indicating whether an animal is a GMO or native variety. He Jiankui, a scientist in Shenzhen, China, attracted significant international controversy when he genetically engineered an embryo and permitted the fetus to be born. Although the paper identified few off-target mutations, i.e. errors in the genetic editing, it is unknown what the long-term consequences are for the child. However, the event foreshadows a potential future role for genetic editing when performing life planning for children. Genomic imprinting is the new fingerprint, allowing anyone to be identified based on their genetic code. It is estimated that information available in public databases is able to identify 60% of the U.S. population. This has implications for law enforcement, identity protection, and more. If you are keen to learn more about genetic engineering, I recommend these videos by Kurzgesagt, a YouTube channel which has made some consumer guides for the topics mentioned in this presentation. If you are interested in keeping up with the scientific papers where these topics are discussed at far greater length, I recommend Google Scholar and the National Center for Biotechnology Information, which index peer-reviewed studies in this field. BioRxiv is a preprint server containing papers before they are expected to be published. The NIH helps to create handy consumer guides for these topics if you aren't interested in the longer scientific journals. I highly recommend the sources in my bibliography. Thank you for your attention.